Okay, welcome back. We are going to go through some different representative drugs and discuss how they work. This is by no means a comprehensive pharmacology class. This is just a very light touch at some different drugs that can affect some specific microbes. The 5 big ways that antibiotics can work. One is by inhibiting the synthesis of cell walls. This is a fabulous way to affect a bacterial population because we as people do not have cell walls. So it is not going to affect us. So something like penicillin is able to weaken those crosslinks in peptidoglycan. And with a weaker peptidoglycan cell wall, the cell will lyse. Meanwhile, we don't have any impact on our cells. Another very good way to treat a bacterial infection is by inhibiting protein synthesis. So if you recall a few chapters ago, when we talked about ribosomes, we discussed how prokaryotic ribosomes have 70 S ribosomes and we have 80 S ribosomes. Our ribosomes have slightly different shapes to them. That means antibiotics can be targeted to a prokaryotic ribosome and they won't affect ours. So we can stop a bacteria from making their proteins, basically shut down the function of that cell without affecting us. One is by inhibiting nucleic acid synthesis. So there are some enzymes that are specific to bacterial DNA replication and transcription. There is also a slightly increased risk that if you affect our own cells' replication and transcription, that we won't be having quite the same cell function. But typically, we are going to target things specific to the bacteria in these processes. One is by injuring the plasma membrane, which will affect the permeability of that cell. So the plasma membrane gets a little leaky and stuff just leaks right out of the cell. And one is by competitively inhibiting essential metabolites, different enzymes within that bacterial cell, especially if we can target an enzyme that is in a bacteria that we don't use. Okay, so here we have inhibiting the cell wall. This would include drugs like penicillins, cephalosporins, bacitracin, and vancomycin. We have inhibiting protein synthesis, so inhibiting ribosomes mainly, chloramphenicol, erythromycin, tetracyclines, and streptomycin. Here we have inhibiting nucleic acid, whether it's inhibiting replication or inhibiting transcription, something like quinolones and rifampin. Injuring the plasma membrane, something like polymyxin B, and inhibiting essential enzymes or essential metabolites, which would be something like sulfanilamide and trimethoprim. Let's go through and talk about these. When we are inhibiting cell walls, mainly we are going to be cross-linking peptidoglycan within the cell wall. Peptidoglycan, again, is this main component of cell walls of bacteria, but not found in our cells. So we don't have a great risk of harming our cells when we harm peptidoglycan. The main targets of cell wall synthesis are penicillins. Penicillins are a group of 50 related antibiotics that all have this core structure. Sometimes it's called a beta-lactam ring, sometimes it's called a nucleus. Just this core structure within penicillin. You can see it kind of makes this tight square that's not very stable, so it'll react with that cell wall to break that cell wall down. Penicillin can be separated into natural penicillin and semi-synthetic penicillin. Natural penicillin is what naturally occurs. It's extracted from the mold Penicillium. However, natural penicillin is susceptible to a drug called penicillinase. So keep these terms straight. Penicillinase, or beta-lactamase, is an enzyme made by some bacteria to give them the ability to be unaffected by penicillin. So if a bacteria makes penicillinase, they are resistant to penicillin. So some microbes make this penicillinase, and it cleaves the beta-lactam ring. It just breaks it apart so that penicillin is no longer functional. So that cell making penicillinase is resistant to penicillin. There's also semi-synthetic penicillin, which is synthetic. It's synthesized in a lab. The nice thing about semi-synthetic penicillin is that it is resistant to penicillinase. So if your little infection says, I make penicillinase, you can't kill me, you throw some semi-synthetic penicillin at it, and that will kill it. So semi-synthetic penicillin is not affected by penicillinase. It has a broader spectrum of activity. So please, when we're talking about penicillin, be able to describe the difference between natural penicillin and semi-synthetic penicillin, and be able to describe what penicillinase is doing. I feel like these words are all so similar: penicillin, natural penicillin, semi-synthetic penicillin, penicillium, penicillinase, but these are all very different words. Please be able to tell me what these different words are without confusing them in your brain. Vancomycin, amoxicillin, cephalosporins, bacitracin, and carbapenem are also drugs that can inhibit cell wall synthesis. Vancomycin is a super potent drug. Really, it's mainly used to kill penicillinase-producing staphylococcus. So these bacteria, especially the Staph aureus, that are resistant, they've developed resistant to everything else. Vancomycin is used to kill off that infection. Typically, it's going to be given as an IV and carefully controlled. It can be toxic to ears and kidneys, though, so it is not the first one you're going to turn to. Amoxicillin is the one you're going to turn to first. It is the most common treatment that's given to children. If you have kids, chances are within the first few years of their life, at some point, they have had amoxicillin. And chances are you have at some point had amoxicillin. Because amoxicillin is able to be cleaved by penicillinase, we are going to combine it with clavulanic acid. Clavulanic acid is going to break down penicillinase. So when you have this bacteria saying, you can't kill me with that penicillin, I have my penicillinase, you throw in clavulanic acid destroys the penicillinase, and then amoxicillin comes in with that punch to knock out the bacteria. So amoxicillin and clavulanic acid are taken together so that they don't have that resistance from bacteria. Clavulanic acid is made from Streptomyces clavulgaris. So again, clavulanic acid is not killing the bacteria. Amoxicillin is breaking down the cell wall. Clavulanic acid is breaking down the penicillinase. Okay. Then we have cephalosporins. Cephalosporins are also able to be used against penicillin-resistant strains. Bacitracin, if you've ever read like triple antibiotic ointment, the kind of stuff you put on when you get like a scratch or something, it typically says it has bacitracin in it. This is a topical application. So as opposed to these, which are IV or taken orally, bacitracin is just spread topically on your skin. That's because your skin does have a lot of gram-positive bacteria on it, and you don't want that to infect a wound. So that little bit of bacitracin will kill off any of your skin flora that could get into the wound. And then carbapenem is another drug that is typically used just when we're dealing with drug-resistant infections. It does have a broader spectrum because it can treat a wider range of bacteria, but it does still inhibit the synthesis of those peptidoglycan cell walls. Okay, we have a couple of bacteria categories that are slightly different in their cell walls. If you remember talking about mycobacteria, our acid-fast bacteria, they have that waxy stuff called mycolic acid in their cell wall. So they're going to stain as being acid-fast. The way we can treat them is isoniazid and ethambutol. So isoniazid is going to inhibit mycolic acid being made, and ethambutol is going to stop mycolic acid from being put into the cell wall. So these are really good for killing mycobacteria, especially Mycobacteria tuberculosis, which is what causes tuberculosis. Next, let's get into inhibiting protein synthesis. So here is our little ribosome with the messenger RNA, the tRNA coming in, building a little polypeptide for this cell to grow. However, if we throw in chloramphenicol, it is going to bind to this 50S portion of the ribosome and stop that peptide bond from being made. So no more amino acids can be incorporated. You stop making proteins. Tetracycline is going to come in here and affect how tRNA is going to affect the messenger RNA. So you're not going to end up with functional translation happening. And streptomycin is going to change the shape. See how it made this little bump here? So you don't have a very effective codon being read here. So messenger RNA gets read in all kinds of crazy ways. You don't get a functional protein. So chloramphenicol, tetracyclines, and streptomycin are all going to inhibit the synthesis of proteins. Let's see. Macrolides, streptogranins also are able to prevent synthesis of 7DS ribosomes. Macrolides, something like erythromycin. Was that on this? Okay. Erythromycin that stops amino acids from being added. Streptogramins also bind to the 50S portion and stop amino acids from being incorporated. And then oxazolidinones are going to stop the ribosome from even being made. So they're going to stick onto that 50S portion of the ribosome and stop it from binding to the 30S portion. So you don't even make a ribosome at all to begin with. When we're talking about inhibiting nucleic acid synthesis, rifamycin is used to inhibit the synthesis of RNA. So it is mainly used to treat tuberculosis. Quinolones and fluoroquinolones are used to inhibit the enzyme gyrase. If you remember from our discussion in Chapter 8, hopefully you do, we just had this discussion, gyrase is unique to bacteria. We don't use it in eukaryotic cells. So by inhibiting gyrase, the bacteria are not able to make their replication fork, and they're not able to replicate their DNA, so they stop growing. It doesn't necessarily stop them from living as what's there, but it stops them from producing anymore. It's more bacteriostatic. Then polymyxin B and isoniazid are used to inhibit the plasma membrane of bacterial cells. Polymyxin B will make the membrane permeable. That means things can leak out. So you don't have a good functional cell if it can't even keep its stuff together. Isoniazid is going to stop getting those fatty acids put into the membrane, so you can't make a good membrane. So the cell's not able to grow at that point. And then this one I think is just fascinating in how it works. Sulfonamides are going to be a competitive inhibitor for an enzyme called PABA. PABA is an enzyme that bacteria use to make folic acid. Now, people, we need folic acid too, right? It's an important vitamin that we need. But we don't make our own folic acid. We have to eat it. You might get it from eating enriched cereals, from eating spinach and leafy greens. You might get it just from taking a daily multivitamin. But you have to eat it somehow. You're not going to make it. So if you inhibit this enzyme the bacteria are using to make it, the bacteria stops being able to make folic acid, and they just fall apart. They can't figure out how to exist if they're not making folic acid. Meanwhile, you're still eating your spinach, so you're still getting the folic acid and growing just fine. Sometimes you'll see it as TMP-SMZ, which means trimethoprim and sulfamethoxazole. This is actually a drug that we'll get to try out in lab to test how it works, which is going to just act as a competitive inhibitor. It just stops this enzyme from making folic acid. Okay, so now you can tell me what cellular function is inhibited by tetracyclines. Of the 3 drugs often found in over-the-counter antiseptic creams, this includes polymyxin B, bacitracin, and neomycin, which has a mode of action most similar to penicillin. And one of the most successful groups of antibiotics targets the synthesis of bacterial cells' walls. Why does this antibiotic not affect mammalian cells? Okay, we've still got a little more to talk about. We're going to talk about some resistance and what happens if you mix drugs together. So we've got a couple more lectures for this chapter, and then you'll have your second exam after chapter 20. All right, I'll see you later. Let me know if you have any questions. Bye-bye.